Hello, everyone. Today, it's my pleasure to introduce an interpretable machine learning model for predicting 5-year survival in breast cancer based on integration of proteomics and clinical data. In this study, 773 untreated breast cancer patients from all over China were collected and followed up for at least 5 years. We obtained clinical data from 773 cases, RNA sequencing data from 752 cases, and proteomic data from 271 cases. We used 5 different data combinations to develop and train the model, and then compared the performance of the different feature combination models using 5 different algorithms. By optimizing the number of features, select the most important subset from a large number of features. Finally, an optimal model containing 13 features was determined. In order to make the model transparent and trustworthy, we used advanced interpretability techniques including SHapley and KAN. In addition, we will encapsulate the explanatory model into a user-friendly network tool for clinical doctors to use, enabling real-time prediction and result visualization. Finally, to further confirm the biological basis and clinical relevance of the model, we conducted immunohistochemical verification. Among the 5 characteristic combinations that predict the 5-year survival rate of breast cancer patients, the performance of the proteome and clinical data, AUC equals 0.814, is significantly better than the other combinations, which proves the necessity of multimodal fusion. The feature optimization process table shows the process of simplifying features from 177 to 20 using a 3-step screening method. Then, 5 machine learning algorithms were used to evaluate these 20 features, and DNN performed the best. Afterwards, we further optimized the features using SHapley values. As the number of features increases, model performance improves, but tends to stabilize after reaching 13 features. Therefore, we selected the top 13 features for further analysis. To elucidate the efficacy of our ultimate model, we employed SHapley to explore the contributions of various features. The results showed that significant features contributing to the prediction were MPOSPH10, EGFR, ARL3, KRT18 and lymph node positive and so on. MPOSPH10 is the most important feature, while ACOX2 has the lowest importance. The strength and direction of the relationships between various features are shown in the figure. SHapley local explanation elucidated how individual characteristics impacted the probability of a 5-year survival prediction for each patient. The no 5-year survival and the 5-year survival for specific breast cancer patients were deduced by visualizing the decision-making of the SHapley values of the 13 features. We validated and interpreted the optimal model integrating 13 proteomic and clinical features using KAN to further enhance the transparency and interpretability of the model. ROC curve analysis demonstrated an exceptional classification performance. Visualize the KAN network topology and clarify the output results of the 5 survival prediction model for breast cancer patients. Furthermore, fitted function analysis identified that MPOSPH10 and tumor size as key contributors to the models. Prediction outcomes, exhibiting significant linear correlations between key features and prediction outcomes. To enhance the clinical application of the final model, we developed an intuitive web application based on the Streamlit Python framework. Clinicians or patients could input the values of 13 key characteristics on the left interactive interface. The 5-year survival prediction result could be real-time assessed by the SHapley force plot on the right. Finally, we investigated the protein expressions of the 9 proteins of the prognostic predictive model via HPA database. Immunohistochemical staining analysis revealed that the 7 proteins shown in the figures were differentially expressed in breast cancer compared to normal tissues. The interpretable multimodal model integrating proteomics and clinical data constructed in this study has shown a strong performance in predicting the 5-year survival rate of breast cancer patients. It goes beyond the traditional black box model and uses SHapley and KAN methods to explain and reveal identified protein markers, especially MPOSPH10, which represents promising prognostic biomarkers and therapeutic targets. The developed online prediction tool enables the model to be conveniently used in clinical practice, assisting doctors in personalized prognosis assessment and treatment decision-making.